It is absolutely my pleasure to welcome everybody this morning to our next Partners, or MGB rather, Asthma Center Grand Rounds, and to welcome today's speaker, Bruce Levy. Obviously, Bruce does not need a lot of introduction to this group, but he is the Parker B. Francis Professor at HMS, the Chief of the Division of Pulmonary and Critical Care, and the Chair of the Department of Medicine. Scientifically, Bruce Levy has made major contributions to really defining and characterizing the biologic effects of endogenous anti-inflammatory mediators in asthma and in lung inflammation. This includes understanding the several families of SPMs, or specialized proresolvin mediators. In his clinical research, he is the PI of the SARP-IV. This iteration is immunometabolic phenotypes in adult severe asthma and disease progression. And today he's going to give us an update from the Severe Asthma Research Program, potentially on that theme. Thanks so much, Bruce, for being with us.
Thanks, Nora, and hello, everyone. It's really nice to be able to be with you this morning. I'm going to give the update from the Severe Asthma Research Program 4. So let me put it in. Nora, does that look okay?
Now it does, yeah.
Wonderful. Okay, good. I only had the Partners Asthma Center logo to put here, but we do need to upgrade that to MGB, update it. I guess not upgrade it, update it. So I do have a few disclosures related to some equity interests, some consulting work that I've done in the past, and funding from the NHOBI and some industry partners that's certainly relevant for today. Okay, so what I thought I would do is kind of start with the Severe Asthma Research Program, SARP for short, the hypothesis and aims in this most current iteration, and then provide you with a little bit of a smorgasbord of topics where there's been now several publications that have come out in the last 5 years or so related to these topics that I think are broadly relevant as we think about severe asthma airway structure and function, mucus plugging, respiratory tract granulocytes, and this NETosis phenomenon, corticosteroid responsiveness, metabolic dysfunction, insulin resistance, asthma genetics, and genomics. I could go into great depth on any one of these topics, but I thought I would give this broad audience kind of a broad swath of what's been published recently as part of SARP. So the definition of severe asthma that's used in SARP is shown here. It's basically a lot of steroids, either by inhaled corticosteroids or oral. And then one of the following that either occurs despite being on steroids or would occur if you started to reduce treatment. And that's a lot of symptoms, a couple exacerbations in a year, at least one severe exacerbation, or limited lung function, less with an FEV1 of less than 80%. So the SARP-IV main hypothesis is shown here. Extensive longitudinal and immunometabolic phenotyping will advance our understanding of the heterogeneous mechanisms for severe asthma and inform future therapeutic development, clinical management strategies. Specifically, comprehensive phenotyping will reveal distinct biological mechanisms and clinical outcomes for severe asthma patients with persistent T2 inflammation, non-T2 disease, and effective resolution. And so to test this, SARP is engaging in a national multi-center collaborative study, mechanistic translational approaches, and there are 4 specific aims to perform this work at molecular and cellular origins of the immunometabolic phenotyping and relationship to disease progression. SARP is a collaborative, this is from SARP-III, it's actually not even updated to SARP-IV, so it's actually been expanded even further in terms of sites across the nation. And so none of what I'm sharing today comes from any one individual lab. It's really come from this collaborative approach across the nation in an attempt to better understand severe asthma. Our goal was to recruit 400 asthmatics as part of SARP-IV, and we are now fully recruited in our asthmatic participants. And we're nearly fully recruited with our controls. We have just a small number, a handful of controls that are still to be recruited. The only reason for the delay there is we're trying to make sure we do bronchoscopy on the remaining healthy controls. So we want to have as robust an array of biospecimens as possible for mechanistic investigation. So the four aims are first this to look at the interplay between epithelial function, airway immune cell activity as drivers of severity and poor asthma control. Second, to look at this relationship in more depth between metabolic dysfunction and non-T2 inflammation and persistent severe asthma. Thirdly, to look at resolution mechanisms that may be defective in severe asthma in persistent forms of both T2 and non-T2 disease. And then fourth, apply unbiased systems-based approaches to uncover unsuspected mechanisms of disease and predictors of disease progression. So as I kind of delve into this, I just tried to kind of aggregate recent publications into themes that have emerged. And a lot of the publications that are coming out now relate to SARP-III. So SARP-III and SARP-IV combined, we have a couple of hundred people in SARP-IV that were also part of SARP-III. So that means we have a cohort of people we've been following now for over a decade as part of this program. So really, this is the longest cohort for severe asthma that is kind of in the public domain, if you will. And we also, of course, have cross-sectional analyses that we're doing. So in terms of airway kind of structure and function, just to, this is just for perspective. Nizar Jorjur from Wisconsin kind of helped to lead this kind of establishment of what SARP had contributed about 10 years ago in looking at airway structure and function. Excuse me. And at that point, you can see about 1,600 patients have been characterized and about 583 with severe asthma. You can see that in terms of lung volumes and FEV1 shown in the blue and the residual volume shown in black, as you might expect, severe versus non-severe versus normal. And then in many different ways, mild across the top, severe across the bottom. You can see characterization emerging from SARP related to studies with quantitative imaging by chest CT and also airway investigation here by bronchoscopy. Sputum's collected, peripheral blood's collected. So it's a pretty broad array of biospecimens. Of course, the early iterations of SARP looked at clinical phenotyping with the goal now in the more recent iterations of SARP to become a little more organ, cellular, and molecular. These 5 clusters had some clinical phenotypes associated with them. The first 2 more mild asthma, mild allergic, mild moderate allergic, and then the last 3, clusters 3 through 5, more severe asthma, either older onset, variable atopic, or with fixed airflow obstruction. And you could see that they've been, they each have some clinical features that are listed. I would refer you to this review article for more information. So because of the wonderful work by George Washko and colleagues, I don't need to introduce this group to the incredible value of quantitative CT analyses. But as it relates to those clusters, recently, this came out in this analysis of over 450 participants in SARP. And looking at several different parameters on chest CT, And lung expansion was found to be about 30% smaller in those with severe asthma compared to participants with more mild asthma. And in the image, you can see the both coronal and sagittal CT images, cluster 1 being the first 2 columns, cluster 5, the other extreme being the last 2 columns. And you can see across the top, even at grayscale, you can see differences by this DPM mapping, the Jacobian mapping, and by this anisotropic deformation index, kind of the lateral pulling of the airways. You can see pretty significant changes from cluster 1 to cluster 5. So airway morphometry and air trapping can be identified and aligns, if you will, with these clinical phenotyping clusters. So one question that has, I think, become much more clear with the study of the CT of severe asthma is addressing this type of a question. Is it severe asthma in the airways more of a uniform involvement of the airways, or is it more spotty? Is it more like colitis or more like Crohn's disease? And a lot of this work kind of focuses in on the mucus plugging story. So I'm going to incorporate that into this section. So this interesting study was published just the last couple years from other group. And in the yellow there, in the image on the left, we're highlighting an airway with a mucus plug, kind of a long mucus plug on it. The orange arrowhead is the partner vessel. And so you can see these mucus plugs in chest CT, and this is separated by 3 years in time. And what you can see is that that mucus plug is still there in that same airway, in that same segment. And if you look by Sankey plot at baseline out to year 3, those who had a plug at baseline largely had a plug in that same segment at year 3. And if you didn't have a plug, you kind of didn't at year 3. So interesting that these plugs can persist in asthma. To look at the relationship between the plugs and air trapping, and this was just published in December, a novel air trapping scoring was developed by the team. And in this light blue, you can see the minimal, in the yellow, more limited air trapping, and then widespread is in red. And you can see that it's judging these different segments in the lung on CT imaging when we look at both inspiration and expiratory films. So if you look at age, you can see more widespread, a relationship with more widespread airflow trapping. You can see more severe FEV1 drops. And then you can see the relationship in males and the relationship to exacerbations. And so with a lot of the, with age, with FEV1, with exacerbations, airflow trapping can be mapped by the score. If you look at the top here on the left, we're looking at blood eosinophilia, less than 300, greater than or equal to 300. So the air trapping segment score is higher with higher blood peripheral eosinophilia. And in panel B here, this is looking at sputum eosinophilia with a cut point of 2%. You can see that airflow trapping is higher with more sputum eosinophilia. In the bottom, it's looking at the percent of participants with these different conditions, minimal, limited, and widespread. And at the higher eosinophils, you can see that there's more participants with widespread airflow trapping. When you relate this to mucus plug, there's this score for mucus plugging is higher with the higher air trapping segment score. And you can see about this pertains to about 40% of participants. So there's a relationship between these plugs and airflow trapping. And this just came out last month in JCI Insight. You can actually map this now down to the branch points of the different airways and the airway generations. You can generate almost like it looks like a family tree of where there's a plug here in red and the airway generation. And from trachea on out, you can see the number of plugs in by airway generation, and you can see that the predominance are in generation 6 and 7. And if you look at the by Spearman coefficient, the airway generation and the relationship to FEV1 or FEF 2575, if the plug is in a more proximal location up to about generation 7, there's a pretty significant drop in FEV1 and FEF 2575. And then there's a kind of a dose-dependent impact as you move out more and more distally. So it looks like proximal airways are consequential for airflow obstruction. And then you can see this also by functional imaging with MRI. This came out in 2022, looking at MRI and the segmental ventilatory defect here, and by with helium MRI. And then if you look at the CT, you can see that there's a mucus plug sitting right there. And if you look at those by MRI, the segmental ventilation defects, and whether or not a mucus plug is related, it's much more common that those are related. Now, it's not a universal finding. There's certainly segmental defects where there's not mucus plugging, but you can see the impact of that plug on hyperpolarized helium MRI ventilation defects. So to summarize this section of the talk, mucus plugs in asthma are more common in severe asthma, can persist in the same airway segment over years, are associated with eosinophilia, and when proximal, are associated with airflow obstruction. So in this next section, I was going to talk about some recent findings related to airway type 2 and non-type 2 inflammation in severe asthma. Of course, this very nice review from Guy Purcell and Gerald Koppelman showed T2 high asthma in the red there and T2 low asthma on the right in the greenish color. Lots of information, and SARP played big roles here in characterizing the T2 high asthma. Of course, we've got many therapeutics now in that space. Much less information related to T2 low, and Melody, just in our last asthma grand rounds, detailed some current thinking around T2 low asthma. And really the players that she focused on, and I'll just mention in terms of SARP contributions to this database, neutrophils, Th1 cells, and Th17 cells. So in the collected sputum from patients in SARP, if you were to characterize it by predominantly T2 high in the top, highly variable, sometimes T2 high, sometimes not. And then in the bottom, predominantly T2 low asthma patients. And you looked at their sputum eosinophilia, you can see that there's some concordance over a 3-year span in the sense that the T2 predominantly high kind of stay high. There's with more than 2% eosinophils. And those in the predominantly low kind of stay predominantly low with less than 2% eosinophils in their sputum. But there is this group that's kind of all over the map, up and down in terms of sputum eosinophilia. And if you look at asthma exacerbations in a year, in the past year, in this dark black circles on the top, that is those with this highly variable eosinophilia. And so a little bit more common exacerbations in that group relative to the ones that stay persistent, either high or low. And then if you look by cellularity at the FEV1, in this kind of black diamond with a solid line are those individuals with both high sputum eosinophils and high sputum neutrophils. And those are here at the bottom over this 3-year period of time with the lowest pre-bronchodilator FEV1. So the highly variable eosinophils seem to be more associated with exacerbations, at least trending that way, and then a lot of cellularity related to lower FEV1, lower lung function. Along the T2 low asthma mechanisms, one that SARP has contributed to is this relationship with IL-6 and metabolic dysfunction. Oh, shoot. That is a shame. That slide is not showing up. Well, suffice it to say the IL-6 levels were higher in both a UCSF asthma cohort and in a SARP asthma cohort. And there was a link to a history of hypertension in this story, and suggesting that IL-6 high was related to more severe disease relative to mild individuals, and more commonly a history of hypertension. So here's the data from the IL-6 story, and here's the relationship to hypertension is down here, sorry. And up here, what you couldn't see before was this relationship to IL-6 high. Didn't really matter whether the BMI was less than 30 or greater than or equal to 30. So not clearly linked only to obesity, suggesting metabolic dysfunction. Okay. All right, and then in terms of further details, more recently, the group has published on the left here. I'll focus your attention to exacerbation frequency, with the light blue being no exacerbations in the last year, the darker blue 1 to 2 exacerbations, and then greater than 2 exacerbations in the bottom. And then whether there's a 3-year longitudinal exacerbation phenotype that's either exacerbation resistant in green or exacerbation prone asthma in red. And you can see here in the bar graphs, exacerbations per subject. This exacerbation prone asthmatics are averaging like 7 or 8 exacerbations over the course of a year, relative to the resistant ones, which have almost none. And then you can see there's an intermediate phenotype. Hospitalizations is in this middle bar graph, and then subjects with an ED visit in the right. And in kind of some pathway analyses, with the target being asthma exacerbation, plasma IL-6 and blood eosinophils were our primary exposures of interest. You can see the relationship to some of these other features. And in the bottom, the predicted number of asthma exacerbations over a 3-year period of follow-up by plasma IL-6 levels was highly significant. More IL-6, more asthma exacerbations. And this, you can see how blood eosinophil cell counts perform in a similar way. Also highly significant, but very similar predictive value as plasma IL-6. Well, this is a work from Melody that she shared with you at our last grand rounds. So severe asthma subjects have increased BAL neutrophils, interestingly, and then the NK cells are decreased in the BAL in asthma. These are CD3 negative NKp46 positive cells. And here they are in healthy control, non-severe and severe asthma. So in both non-severe and severe asthma, they're decreased. When you look at the CD4 positive T cells, those are increased significantly in severe asthma BAL relative to non-severe asthma or health. And we were limited to actually formally, in our ability to formally characterize them as Th1, Th2, Th17, but we use the chemokine receptor signaling to kind of impute what might be a Th1 enriched, Th2 enriched, or Th17 enriched population. And when you do that, you can see that here in the red, looking at Th1 enriched, Th17 enriched, or you see both of these are higher in severe asthma by number. And we didn't see a skewing that was clear with Th2 enriched. The Th1 CD4 positive T cells in particular have this inverse relationship to FEV1, suggesting an association with more limited airflow obstruction. or limited lung function. When we looked at the ratio of the Th1 cells to NK cells, or Th17 cells to NK cells, or even just neutrophils to NK cells, what we can see in the severe asthma cohort is that this is higher, this ratio is higher in all of these three cases. So severe asthma is distinguished by a decrease in BAL NK cells relative to the leukocyte effectors of non-T2 inflammation. We'll first look at the Th1 story in a little more depth. So this is from Anurad Ray from the Pittsburgh group, and this was published in JCI. What was found in the SARP cohort was interferon gamma positive CD4 T cells were higher, as were the IL-17A positive CD4 T cells, also higher in severe asthma in these dark circles relative to the open circles in mild to moderate asthma. What was impressive in terms of the mediators, though, was that interferon gamma was significantly up in severe relative to mild to moderate asthma. Not as convincing a story for IL-17A levels in this cohort. And while IL-5 was not higher, IL-13 was significantly higher. And this profile was in about 35 to 40% of severe asthma patients. The group went on to look at what might be downstream of interferon gamma, and what popped for them was CXCL10, shown here in the red in severe asthma. Here's the blue, mild to moderate asthma, and healthy controls. And this relationship between interferon gamma mRNA and the BAL relative to CXCL10 mRNA and the BAL. You can see there's a pretty nice relationship there. So more recently, SARP has done a protein interactive network analysis from BAL fluid of severe compared to non-severe asthma. This just came out in January of this year. And to cut to the chase here, what was found is that the top three proteins for severe asthma relative to non-severe asthma were these three: PDGF, AA, alpha-alpha, fibroblast growth factor 2, and CXCL7, or otherwise known as NAP2. And this, of course, is processed to CXCL4 in the airway. And it's interesting to note that CXCL10 and CXCL4 exert opposite effects on the production of human Th1 and Th2 cells. So this kind of interplay, if you will, between the activation of the T cells and the chemokines in this protein-protein interactive network in the severe asthma airway. Recently, the group has also looked at CCL5 or RANTES, and this is interesting as well for a couple reasons. This appears to be a potential bridge between T1 and T2 inflammation in asthma. And in the top left here, it's CXCL10 expression in sputum and CXCL5 expression. And you can see that there's a very nice relationship there. And if you generate a cut point of CCL high or 5 high or low, you can see that the lymphocytes are higher in the high group. Lung function here in red is negatively correlated. So the higher the CCL5 is, the lower the lung function is by FEV1 on the left and the FEV1 to FVC ratio on the right. And this is related in part to interferon gamma. You can see the nice relationship there, but also the T2 associated cytokine IL-4. There also was a relationship with CCL5 expression. And then on the right here, looking at eosinophils in the blood and FeNO, these were higher in the CCL5 high group. And then in the sputum, eosinophils and neutrophils are higher. So if you look at the proportion of people with CCL5 high expression in eosinophilic, neutrophilic, and mixed granulocytic sputum, you can see that in all cases, they're higher than the CCL5 low. So suggesting that this may be a partial driver of a mix of inflammation in the asthmatic airway. And it raises the potential for drugs such as Maraviroc to potentially have a role, unproven as of yet, role in some patients with severe asthma. So to summarize this section on T2 and non-T2 inflammation and asthma, highly variable. Let me see if I can go back on. See what I'm not sure why the computer did that, but highly variable type 2 inflammation in sputum eosinophilia and increased sputum granulocytes were associated with the most morbidity. Systemic inflammation characterized by being IL-6 high in plasma is associated with T2 low asthma. NK cell dysfunction is associated with decreased clearance of non-T2 leukocytes. And then T1 high asthma is associated with increased interferon gamma and CXCL10. T2 high asthma associated with CXCL7. And then CCL5 may serve as a bridging chemokine for T1 and T2 inflammation with mixed granulocytic inflammation. So moving forward to the next story, it links corticosteroid responses and a neutrophil web. And this story starts from a publication from our group back in 2018 in Science Immunology, where we found that neutrophil cytoplasts induce Th17 differentiation and skew inflammation towards neutrophilia in severe asthma. So just a word about netosis, the generation of neutrophil extracellular traps and cytoplasts. And as neutrophils die, they can undergo a process called netosis. And this is easily studied in vitro with isolated neutrophils. And across the top of this schematic, you can see that what happens is that the cells decondense their chromatin and then kind of emit it. It gets released like a net, breaking up the plasma membrane. And then you have extracellular DNA with a hypercitrinylation of associated histones and many associated proteins. But in vivo, it appears that there's a non-lytic form of netosis where the cells don't die. They actually release this net, but then they reseal to form now an enucleated cytoplast shown in the bottom schematic. This non-lytic netosis has not been able to be studied in vitro, but in vivo, there's clear evidence for this process to occur. So if we look at the BAL fluid and DNA in severe asthma, what we find is that there are some individuals with severe asthma who have high levels of extracellular DNA in their BAL fluid. And if you break this down by neutrophil predominance, this is particularly in the far right here, those with severe asthma and greater than 5% neutrophils in the BAL. And if you do a Western blot looking for this specific hypercitrinylated H3 histone, this is indicative of netosis. This would not happen in just routine cell death. And so you can see in those with high neutrophil counts in the BAL, we were able to find evidence for this hypercitrinylation event. And if you look at the relationship between BAL fluid, DNA, and neutrophil counts, there's a nice relationship. IL-17 and neutrophils in the BAL, also a nice relationship. And then similarly here now, cytoplasts, the presence of these cytoplasts in BAL and IL-17. So it linked this process of netosis to this IL-17 and neutrophilia. In a follow-up to this, in conjunction in SARP with the San Francisco group, led by Mara Lakowicz-Scroggins, who some of you may know from her now working at the NHLBI Lung Division. In sputum, Mara and colleagues were able to find extracellular DNA in asthma in some individuals in high levels. This was not related to sputum eosinophils or a Th2 gene expression mean, but it was apparently related to sputum neutrophils and MPO even more impressively. And you can see in extracellular DNA high in the in panel F that the values for MPO were quite significantly higher than eDNA low or healthy subjects. Moreover, they went on to show that in this eDNA high group, there was evidence for this hypercitrulated DNA, so you can detect mitosis even in sputum granulocyte analysis, looking for extracellular DNA. UBioPred has looked at this and published in JACI on an IL-17 high asthma phenotype with features of psoriasis, actually, from an immunologic perspective. And this was quite in juxtaposition to what they've determined as an IL-13 high phenotype by gene expression. This was looking at whole genome transcriptomic analysis on bronchial biopsy specimens. 22 of 91 patients had the IL-17 phenotype. 9 had the IL-13 epithelial gene signature. And those who were IL-17 high had frequent exacerbations, airway, both sputum and mucosal neutrophilia, decreased lung microbiota diversity, and interestingly high levels of urinary thromboxane B2. And so this suggests that there are at least a subset of people with severe asthma that have an IL-17 high phenotype that may be linked to NETosis. This came out in Science Translational Medicine in the last year, not related to SARP. A group of investigators looked at extracellular trap production and gene expression in neutrophils and found that it could influence corticosteroid responses in asthma. And they ended up focusing in quite distinctly in their gene expression associations on ICS non-responsiveness on this CCL4L2. So it's kind of like MIP1 beta, but it's a distinct chemokine. And in panel C here, you can see in all these three large data sets of gene expression, including CAMP, there was a relationship between CCL4L2 in non-responsiveness to ICS. And then you can see also that the levels of the CCL4L2 were higher in non-responsive subjects relative to responsive subjects. More biology needs to be done to better understand this, but it was a very interesting study and links potentially this NETosis story to corticosteroid responsiveness. In SARP-III, we provided as an evoked phenotype triamcinolone parenterally, both to all subjects, and studied them both before and after the triamcinolone. And in the left in panel A here, in this publication that came out just a couple of years ago from Lauren Denglinger, led by Lauren Denglinger in the SARP group at Wisconsin, looking at FEV1 across the horizontal axis after triamcinolone relative to before triamcinolone, you could distinguish 4 subsets of patients. Those over time, over a 5-year period, who lost lung function at greater than 2% per year, the severe decliners I've highlighted in red, and they're in the graph. You can see the curve there related to parenteral corticosteroid responsiveness. And then mild decline, no change, and even improvement. And in terms of panel B, you can see the predictors of severe decline and exacerbation frequency. You can see the odds ratio, but the lung function response to corticosteroid administration was the most significant predictor of a severe decline in lung function over the ensuing 5 years. And that would be individuals you can see were predominantly those who did not respond to the parenteral steroids, had the most significant lung function loss over the ensuing 5 years. So as part of SARP, we've also done bronchial epithelial cell transcriptional responses to ICS and looked at their relationships to severe asthma outcomes. You can see that there was a kind of a broad gene expression look here. Healthy controls are in the purple, moderate asthma in blue, and those that did not have corticosteroids, those with corticosteroids in panel B are distinguished by a corticosteroid responsive gene signature. So in green, light green there, those with moderate asthma did respond. In the tan color, they did not. You can see severe asthma also has these two cohorts in light red and darker red, more maroon. And if you look at those cohorts, the moderate asthma and severe asthma related to corticosteroid responsive gene expression, you can see in the bottom there, well, the MAR scores are similar, but the FEV1, in particular, severe asthma with low corticosteroid responsive gene expression is lower, most significantly. The FEV1 to FVC ratio is lower, and the symptom score, the ACQ-6, is higher in those individuals. And you can see a pretty broad look at the gene expression signaling in this graph in the bottom left. Those are people with more severe asthma and less corticosteroid responses. And you can see that there's a lot of chemokine and T cell signatures there. And the T cells were more abundant in the bronchial epithelial biopsies in those individuals as well. Interesting lipid signaling is there to some degree as well. You can see in the top, in the middle there, and more even skewed a little bit to the right as the responsive genes to corticosteroids. If you did a concomitant gene expression analysis in peripheral blood, you could find a 7-gene signature, which is shown here in panel E, that actually could predict the epithelial corticosteroid responsiveness with high fidelity. And you can see each of these genes and their expression patterns on high in blue, low in red, epithelial corticosteroid responsive. 6 of them trend in a similar direction, but 1 in the opposite direction. So this is an interesting approach now, looking at some of the mechanisms underlying epithelial cell responsiveness and a link to asthma outcomes. So in this section, I've tried to highlight netosis and corticosteroid responsiveness in asthma. And netosis is linked to non-T2 inflammation, in particular now the Th17 story. Netosis influences corticosteroid responsiveness to some degree. And then this evoked phenotype with parenteral corticosteroids was associated with a lung function trajectory over 5 years. And then bronchial epithelial cell responses to ICS can be associated with severe asthma outcomes. In this next section, I thought we'd talk about insulin resistance and metabolic dysfunction. And this gentleman, you can see, is kind of saying, oh, you might imagine what that's related to. Well, in addition to the IL-6 story, of course, folks here at the Channing have also looked at metabolic relationships in asthma and had this really wonderful paper from Rachel and Jessica Lasky-Sue and Scott and others. And basically, in this nice editorial about the work, where this image comes from, but summarizing a lot of work in this paper, that there were 5 metaboendotypes that were identified and validated, with significant differences in asthma relevant phenotypes, including lung function. Cholesterol esters, triglycerides, fatty acids were among the most important drivers of the endotype membership. So if we look at SARP and the SARP group in some detail as it relates to metabolic function and insulin resistance, this is data that was just published in late 2022 about the SARP cohort. And you can see in panel A, the BMI distribution, all the way from the light blue, less than 25, to the darkest blue, greater than 40. The waist measures, whether they're increased in dark blue, waist to hip ratio increased in dark blue, glucose levels higher than 125, insulin levels higher than 25, elevated levels of hemoglobin A1C. And you can see in all these cases, there's an increase in the SARP population. And then in the calculated HOMA-IR, which is a measure of predictive of insulin resistance, with scores certainly greater than 5 in orange, but also in the transition intermediate zone of 3 to 5. There's a significant number of people in SARP that have evidence for insulin resistance and metabolic dysfunction. If you look at this in all participants in the top here related to lung function, you can see the post-bronchodilator FEV1 and FVC track by HOMA-IR category, less than 3 in blue, 3 to 5 in yellow, greater than 5 in orange. And in the bottom, you can see in BMIs greater than 40, you can see a similar, so kind of irrespective of BMI in a cross-sectional way. And then if you look longitudinally over a 5-year period of time, you can see that the post-bronchodilator FEV1 and FEV1% predicteds were lowest in those with most significant HOMA-IR scores. And in the yellow, the 3 to 5, they actually had the fastest decline in lung function. FEV1 in the top, FVC in the bottom, while the BMI basically kind of stayed the same. But you can see that there is a relationship between BMI and HOMA-IR score. You can pick this up on quantitative CT imaging. Matt Tattersall up in Wisconsin did this analysis that was published recently on skeletal muscle adiposity and lung function trajectory in SARP. And here, looking at the paraspinous muscle density, non-contrast CT imaging, you could take this measure right at the T12 costovertebral junction. And so you can locate and do it the same on everybody, and then take the average. And of course, adiposity is associated with lower Hounsfield units. And what you could see is that both in male and female asthmatic subjects in the pink here relative to controls in purple, there was a change that was significant in the paraspinous muscle Hounsfield units. And when one looked at the FEV1 predicted over time as a change from baseline and unadjusted with clinical adjustments, and then additionally by BMI adjustment, less so in males over time, but in females, there was a pretty strong trend for decrease with more skeletal muscle adiposity. So a decrease in the FEV1% predicted. So in this section on metabolic dysfunction in asthma, high incidence of obesity and metabolic dysfunction in the SARP cohort, measures of insulin resistance were associated with decreased lung function and more rapid decline over time. And then CT measures of paraspinous muscle adiposity are linked to lung function decline over time in women with asthma. In this last data section, I just wanted to highlight a few things that have also come up through SARP related to asthma genetics and genomics investigations, a little bit of a potpourri. Joe Zine and colleagues in SARP have looked at the benefits of airway androgen receptor expression in asthma, and across the horizontal axis is the androgen receptor normalized gene expression. You see men and women and percent of predicted FEV1, the FEV1 to FVC ratio here in panel B, and then the symptom score, AQ-LQ, a significant relationship, positive relationship in all of these. And in the same paper, looking at adverse events, namely asthma exacerbations, ED visits in both the SARP cohort, a cohort from Cleveland Clinic, and an NHANES, just validating what we all see clinically, that the percentage of women, here in parentheses, experiencing these adverse outcomes is greater than the percentages of men in all cases. A group from Arizona has looked at CC16, a protective bronchial express protein, and its relationship to asthma severity and its expression, as shown here on the vertical axis. You can see in a cross-sectional cohort in SARP, there was a link to severity, and then also in the longitudinal follow-up, a link to severity with CC16 mRNA expression in bronchial epithelial cells. Victor Ortega, formerly in Wake, now in Arizona, team in SARP looked at DNA sequencing analysis for the CFTR gene and found cystic fibrosis-associated variants in SARP in a small number, but a significant number of subjects with severe asthma. And if you'll remember back to the mid-90s, there was a paper that seemed to demonstrate protection against bronchial asthma by the Delta F508 mutation with a heterozygous advantage. This story is, I think, still to be completely written. There were some clearly pathologic variants that were identified in this sequencing effort. So it does raise questions about whether we should think about sequencing for CFTR in some of our most severe adult asthma patients. Expression of MUC5AC and MUC5B in the SARP cohort was looked at. Across the top, a relationship with MUC5AC expression, and the bottom, MUC5B expression by RNA in bronchial epithelial cells. And in blood eosinophils on the left, in the middle, sputum eosinophils, and on the right, FeNO, you can see kind of positive relationships, more of these, more MUC5AC, and you can see that it's either flat or negative relationships related to MUC5B. Gasdermin B was also looked at it with other GPI linked proteins that had shown up in an EQTL in the bronchial epithelial gene expression, namely PGAP3 and Gasdermin A. And Gasdermin B, in addition to being associated with asthma susceptibility, also seemed to have at least some relationship to exacerbations. They went on to look at, so here's emergency room or hospitalization, and number of exacerbations and symptoms. And they went on to look at related genes, and there appeared to be an interferon, an antiviral kind of cohort of genes that were downstream of this. So suggesting that Gasdermin B is not only associated with susceptibility to asthma, but also exacerbations. And then the group here with J.C. Cardet looked at RGS2. And this, of course, is part of a regulatory mechanism for terminating bronchoconstrictive G-alpha-Q signaling that drives calcium in human airway smooth muscle cells. And in the panel A here, looking at human airway smooth muscle cells transfected with an siRNA for RGS2, you can see a dose-dependent impact of increasing calcium release in response to histamine. In the non-receptor mediated ionomycin stimulated, no significant differences. So there was a functional change. You can see the expression by Western blot and gene expression in the bottom. And when one looked at the annualized exacerbation rate in non-Hispanic whites, there was an increase in the GG phenotype here for genetic variation. So a link to asthma exacerbations in non-Hispanic whites. So RGS2 promoter variant associates with molecular and clinical phenotype with enhanced bronchoconstrictive stimulation in vitro and higher asthma exacerbation rates in non-Hispanic whites. So I've taken you on a pretty wide-ranging tour of data emerging from SARP. A lot of publications over the last 5 years and more in queue. And I've tried to highlight by themes some pathobiology that's relevant to severe asthma, and airway structure, function, mucus plugging, respiratory tract granulocytes, and netosis, issues and new findings related to corticosteroid responsiveness, metabolic dysfunction, and insulin resistance, and then a few highlights, some recent findings on asthma genetics and genomics. So here we've reviewed published evidence from SARP that anatomic and functional chest imaging is enhancing our understanding of airway structure and function in severe asthma and the importance of proximal mucus plugging. We've further elucidated cellular and molecular immune mediators of T2 and non-T2 inflammation in asthma, provided insights into new relationships between NETosis and corticosteroid responses, highlighted strong associations between insulin resistance and asthma outcomes, and then some genetic variants associated with severe asthma. So comprehensive phenotyping is revealing distinct biological mechanisms and clinical outcomes for severe asthma patients with persistent T2 inflammation and non-T2 disease. Lots of people to acknowledge. This is a village. These are just the names of the leaders at each of these sites. Of course, locally, Elliot, Wanda, and then Justin and Angelus and the whole ARC team have been amazing collaborators. The DCC at Penn State with Dave Major, Brenda, and Kendall are fantastic. They keep us on track. At Arizona, Cleveland Clinic, Stanford, UCSF, Colorado, Iowa, Kansas, Pittsburgh, Wisconsin, Wake Forest, Washington, St. Louis. It's a wonderful team that's been assembled. At NHLBI, Patricia Noel has really been a hero for many years, and now after her retirement, Janing Liu. We have many wonderful patients, many of whom have stuck with us for more than a decade. We're really grateful to them. And locally, Melody Duval and Saisha and Yuan and many other members of the lab have contributed to the story and to our SARP-related activities. And none of this would have been possible really without Chris Vanta, Jeff Drazen, Elliot, and their effort to establish the Asthma Center that we've, of course, all grown to know and love. And lastly, in addition to NHLBI, we've importantly partnered with industry and received funds from the companies listed here in SARP-III or in SARP-IV to help advance the science. So thank you all for your attention. And thank you again for the opportunity to be here to talk with you today about the SARP contributions.
Bruce, thank you so much. That was a whirlwind. There's a ton of questions in the chat, almost none of which we'll be able to get to that people have messaged me. So we'll maybe just ask one or two things. There's a few people messaged me with questions about proposed mechanisms of decline in lung function, particularly if you're measuring something like adiposity, and also how reproducible is triamcinolone in being able to, the lack of improvement in triamcinolone in being able to identify those patients.
So lots of work going into loss of lung function right now, trying to better understand it. I think the physiologic changes are clear. It's certainly not only linked to BMI. So I think it's premature. It would be speculative for me to talk about this, but it is something that we're actively studying in SARP-IV right now. We just don't have any concrete information to share, and I wouldn't want to mislead. As it relates to the triamcinolone, we did that to evoke a phenotype to try to distinguish between difficult to treat asthma from biologically severe asthma. And so I think there's potentially some interesting stories there that could relate to what that evoked phenotype is uncovering, if you will. But it was pretty reproducible. It was a highly significant finding. Wanda and Elliot contributed greatly to that story, and I might refer you to them for more information. People know where I live, know where I sit, so I'm also happy if there's a lot of questions afterwards to try to address them outside the context of rounds.
Great. I think there's a lot of other unanswered questions, but I think we're at time, and to respect everybody's time, and I'm sure you're probably have 10 other meetings in the next 10 minutes, so we'll let it end there. Thank you so much. It was just absolutely wonderful tour de force.
Thank you. Thanks, Nora. Thanks for the invitation. Thanks, Chris. Have a good day, everyone.